In this edition, we spotlight on apoptosis. Apoptosis is an important biological process and its defection has been implicated in an extensive variety of diseases. This time, we focus on the molecular mechanisms, relationship with the tumor, and detection methods. Apoptosis is a form of programmed cell death, or cellular suicide. It is different from necrosis, in which cells die due to injury. Apoptosis is an orderly process in which the cell's contents are packaged into small packets of membrane for garbage collection by immune cells. Morphologically, apoptosis is first characterized by a change in the refractive index of the cell followed by cytoplasmic shrinkage and nuclear condensation. The cell membrane begins to show blebs or spikes, protrusions of the cell membrane, depending on cell type, and eventually these blebs and spikes separate from the dying cell and form apoptotic bodies. Apoptosis and necrosis occur under different circumstances and involve different steps. Simply put, necrosis is messy and causes an immune response of inflammation, while apoptosis is tidy and splits the cell into little parcels that can be taken up and recycled by other cells. Cells that undergo apoptosis go through a different and much more orderly process. They shrink and develop bubble-like protrusions, technical name, blebs, on their surface. The DNA in the nucleus gets chopped up into small pieces, and some organelles of the cell, such as the endoplasmic reticulum, break down into fragments. In the end, the entire cell splits up into small chunks, each neatly enclosed in a package of membrane. When cells are damaged by harmful factors, such as injury or toxic chemicals, they usually spill their guts as they die. Because the damaged cell's plasma membrane can no longer control the passage of ions and water, the cell swells up and its contents leak out through holes in the plasma membrane. This often causes inflammation in the tissue surrounding the dead cell. The following table shows the morphological features of apoptosis and necrosis. There are two major types of apoptosis pathways, each of which illustrates an important point about how apoptosis is triggered and why it is useful. In the extrinsic pathway, a signal is received from outside the cell instructing it to commit programmed cell death. This may occur if the cell is no longer needed, or if it is diseased. Like many pathways for bringing about complex changes in a cell, the extrinsic pathway to apoptosis involves many steps, each of which can be upregulated or downregulated by gene expression or by other molecules. The intrinsic pathway is triggered by stress or damage, which can lead the cell to apoptosis, include damage to its DNA, oxygen deprivation, and other stresses that impair a cell's ability to function. In response to these damages or stresses, the cell decides that its continued existence might be dangerous or costly to the organism as a whole. It then activates a set of proteins called BH3-only proteins. In some cases, a cell can pose a threat to the rest of the body if it survives, which may be the case for cells with DNA damage, precancerous cells, and cells infected by viruses. When a cell's DNA is damaged, it will typically detect the damage and try to repair it. If the damage is beyond repair, the cell will normally send itself into apoptosis. When cells have DNA damage but fail to undergo apoptosis, they may be on the road to cancer. Sometimes, precancerous cells manage to duck both internal and external cues that would normally trigger apoptosis. This allows them to divide out of control and accumulate mutations, changes in their DNA. A number of methods have been developed to detect apoptosis. These methods are based on the changes in the different apoptotic events, including plasma membrane alterations, mitochondrial changes, changes in the cytoplasm, DNA modifications and so on. An X and V apoptosis assay is based on the measurement of the loss of plasma membrane asymmetry. The analysis is typically by flow cytometry. An X and V binds to phosphatidylserine, which migrates to the outer plasma membrane in apoptosis. Pair NX and V with a membrane-impermeable dye like pi to distinguish between intact, apoptotic, and necrotic cells. Caspases are synthesized as inactive precursors, which are activated by proteolytic cleavage to generate active enzymes. The activation of caspases is a common and critical regulator of the execution phase of apoptosis, triggered by many factors, including treatment with radio and chemotherapeutic agents. Activation of caspases can be examined through a variety of methods: colorimetric, immunoblot, or immunohistochemical. Mitochondrial membrane potential, MMP, is an important parameter of mitochondrial function that has been used as an indicator of cell apoptosis. Several cell membrane permeable fluorescent dyes, such as rhodamine 123, Diox6, JC1, TMRM, and TMRE are currently available to measure changes in MMP. Mitochondrial cytochrome C has been found to have dual functions in controlling both cellular energetic metabolism and apoptosis. Through interaction with apoptotic protease activating factors, APOF, cytochrome C can initiate the activation cascade of caspases once it is released into the cytosol. A hallmark of late apoptosis is extensive genomic DNA fragmentation that generates a multitude of DNA double-strand breaks with accessible 3-hydroxyl groups. Tunal assays identify apoptotic cells by the terminal deoxynucleotide transferase-mediated addition of labeled deoxyridine triphosphate nucleotides to the 3' end of DNA strand breaks that are subsequently visualized depending on the introduced. Apoptosis is associated with the fragmentation of chromosomal DNA into multiples of the 180 to 200 BP nucleosomol unit, known as DNA laddering. In DNA laddering assay, small fragments of oligonucleosomol DNA is extracted selectively from the cells whereas the higher molecular weight DNA stays associated with the nuclei. The isolated DNA is separated by electrophoresis and visualized using ethidium bromide. In short, apoptosis removes cells during development, eliminates potentially cancerous and virus-infected cells, and maintains balance in the body. If you have any needs, please feel free to contact us.